Today, we'll be discussing on optimization, a case study of a category factor. Before we continue, make sure you click the subscribe button and please share our link. So we have to talk about what a category factor is. Category factor is a quantitative variable. Can be a factor, can be a variable. This means that this type of variable cannot be quantified. We have two types of category factor. We have nominal and we have ordinal factor. Nominal variable is the one that describes a category without a particular order. Example, type of biomass. It can have different levels like rice hulks is a type of biomass, corn cob, bagasse, etc. We cannot say that rice hulks was listed before corn cob because it is more superior. That's why you say it doesn't have a particular order. But when you talk about ordinary variable, there are those values that are defined by an order relation between the different categories. Example, behavior. Behavior can have different levels like excellent, very good, good, etc. Here we can say that excellent was listed before very good because it's better. So that means it has a particular order duration. Ordinal has a natural ordering that is limited because we do not know by how much excellent behavior is better than good behavior. So it's limited. It cannot really tell you, look at how much this one is better than the other one. This work shows how to analyze optimization work that involve category factor using post-banking design BBD. The case study we have here is the adsorption of dye using 4 different adsorbents. Please, this result is solely for this demonstration and may not represent the true results. The 4 adsorbents were prepared for 2 different biomass. We have spent grain, we have hamburger seashell via 2 different methods, 2 different activation methods. We have conventional, we have microwave methods of activation, giving us 4 different adsorbents. Conventional prepared CPASG is conventional prepared adsorbent from spent grain. CPAHS is conventional prepared adsorbent from hamburger seashell. MPAHS is microwave prepared adsorbent from hamburger seashell. MPASG is microwave prepared adsorbent from spent grain. The optimization tool we are using is Banking Design BBD. It's a type of RSM. We have 2 types of RSM. We have CCD, we have BBD. You can equally look for our work on RSM and get more information on this. BBD is more economically efficient in terms of number of required runs than other type of RSM. We have many factors. When we have many factors like the one we have here, BBD will be the best option. BBD is equally useful in avoiding experiments that will be performed under extreme conditions for which unsatisfactory results might occur because we have only 3 levels, so the number of runs will really be smaller, unlike the CCD that has 5 levels. Factors are level for the adsorption process. Here we have 5 factors. Out of these 5, we have 4 that are numeric, which are temperature, pH, dosage, and initial concentration. Why type of adsorbent is a category factor? We have 4 levels of this category factor here. We have CPASG, CPAHS, MPAHS, and MPASG. This is design matrix generated from the number of factors with the response entered. This response is removal efficiency that was generated after performing the experiment. You can equally do for work on designing your experiment so that you know how to generate this design matrix. The total number of experiments was 116 because the level of category factors, in this case you have 4, multiply the total number of experiments generated from the numeric factor. The experiments were performed according to run order, but was analyzed according to standard order. This is analysis on variance table. It displays the significant terms with their p-values. From the table, the model F value of 292.39 implied the model was significant, which means we have less than 0.01% chance that the large value of this F value could occur due to noise. Every term that has probability greater than F, less than 0.05, indicated model terms were significant. From the table, it was observed that the linear effect of pH, temperature, initial dye concentration, dosage, and adsorbent types were significant. Equally, interaction effect of pH and temperature, which is AB, pH and initial dye concentration, which is AC, pH and dosage, which is AD, temperature and initial concentration, which is BC, initial concentration and dosage, which is CD, were significant. Equally, quadratic effects of temperature, which is B squared, and dosage, which is D squared, were equally significant. Please, you can equally go through our lecture on principles of ANOVA and how to determine the significance of your factor so that you can get more information on these terms on the ANOVA table. Model equation involving category factor. Model equation involving category factor for more than 2 levels are very difficult to analyze. Although I told you that category factors are not quantifiable, but they can appear as a number in a set of data. The number have no mathematical meaning because they can neither be added together nor subtracted. Correspondence between these numbers and categories is established during data coding. Like that category factor we talk about behavior, it can be represented as having levels 1 to 4. 1 means very good, 2 means bad, 3 means good, 4 means excellent. So in regression, these category factors are represented by a dummy variable. These dummy variables are 0 if that type is not present in that run, and 1 if it is present. Therefore, in this case study, where we have 4 levels of category factors, the dummy variables will be 1, 0, 0, 0, 1, 0, 0, 0, 1, and minus 1, minus 1, minus 1. This is the table showing the factors and levels. E means category factor with 3 runs, 1, 2, 3. That's E1, E2, E3. That means run 1 has only CPASG, that's 1, 0, 0. Run 2 has only MPASG, which is 0, 1, 0. Run 3 has only MPAHS, which is 0, 0, 1. CPAHS, which is the last one, has minus 1, minus 1, minus. It serves as the reference level in which every other absorbent times were excluded. Equation 1 is model equation generated in coded form. Because we have 4 levels of category factor, this model equation can be seen as a 4 model equations. 1 comprising of E1 with all its interactions. Just include everything on that equation 1 except E2 and E3. So 2 comprises E2 with all its interactions. The same thing, include everything in equation 1, excluding E1 and E3. And 3 comprises E3 with all its interactions. The same thing, include everything in equation 1, excluding E1 and E2. As equation 4 bears everything except E1, E2, and E3. This equation serves as a reference equation where CPASG, MPASG, MPHS were absent except CPAHS. This equation can be used to predict the response, in this case removal efficiency. You can equally look for a lecture on response prediction using model equations so that you understand this aspect very well. Please don't forget to click the subscribe button and share our link. Remember, the aim of this work is to obtain the optimal condition for the adsorption process. This table presents the optimal condition listed according to the category factors level. The predicted values are the removal efficiency as generated by the model equation, while the experimental value we are gotten after repeating the experiment are the said optimal condition. The essence of the experimental value is to validate your model equation. You can see that the percentage error gotten was quite small, which shows that there was little difference between the between that predicted by the model and that gotten from the lab. Please don't forget to share our link and click the subscribe button. God bless you. Thank you.